Hi Year 12s, Mr. Lim here again, and it is the fourth video on proteins tertiary structures number 1. So we're learning about tertiary structures, and we're going to be learning about disulfide bridges and hydrogen bonding today. Alright, so once the secondary structures are built, so that's the alpha helices and the beta pleated sheets, further modification to the proteins can occur to ensure that it is the correct shape, because remember, it is all about the shape. Tertiary structure of a protein is the final folded shape of the protein. This overall shape is held together by many, i.e. thousands, of small interactions between side groups of amino acids in the chain. Okay, so in secondary structures, the interactions are between the peptide links of the amino acids. Now we are talking about the interactions between the side groups of amino acids, so not the peptide links, and that's the differentiation between tertiary and secondary structures. Right, so diagram of the tertiary interactions. Okay, so this is just a couple of examples of them. Right, so there's the line of amino acids, and then you have a number of amino acids there. Right, and then their side groups stick out of the chain, and then they can interact with other side groups, and then cause it to bend and fold into a particular way. All right, so let's have a look. 5 types of these interactions: disulfide bridges, hydrogen bondings, dipole-dipole. There's not actually much on dipole-dipole. And then dispersion forces and ionic interactions. Okay, if too many of these interactions break, because remember, they're being held together, the only reason why it's in that shape is because they're being held together by these interactions. If you break all those interactions, the protein will unfold and then for no longer have the shape necessary to function. Okay, because it's about the shape that it functions. Okay, so disulfide bridges, what are they? They form when two cysteine amino acids in different locations are brought together by folding, and then the hydrogen connected to the sulfur is removed, and then the two sulfurs atoms are covalently bonded to each other. Okay, that's a redox reaction, but let's have a look. This is a cysteine cysteine amino acid. Okay, so CH, and I'm just going to draw the COO over here. Let's just draw the CO there and the NH over here. Okay. And that would be bonded to the next one, that would be bonded to the next one, and then there's a side group here, CH2, and then an SH, and then here's another one, another part of that chain. Okay, so remember, that chain can go all the way back here, and maybe it comes back here, and then you're going to be ending up with a COO over here, right, followed by a CH and maybe a CH2 there. Not really, maybe, definitely. And then an S and a H here. Okay, and then it's continued on with an NH, something there. Okay, so remember, interactions with side chains, not the peptide bonds. Okay, what happens is that those two hydrogens get removed, and a brand new covalent bond is formed between them. Okay, so not, like a ionic bond or whatever, it's a covalent bond. Okay, so there's your diagram. Okay, and there's another diagram showing that how they can hold the protein together. Okay, because this is a covalent bond, it is very strong bond, and therefore it takes a little bit more to break, but it holds these proteins in place. Okay, so it's a covalent bond between the two sulfurs. Yep. Okay. Hydrogen bonding. So remember, we dealt with hydrogen bonding of peptide links in secondary structures. Now we're dealing with hydrogen bonding in tertiary structures, which will be between two side groups of amino acids. Okay, so it's super important to remember tertiary structures, side groups. Okay, different to secondary structures because the hydrogen bonding is between atoms of the side groups instead of the peptide link as in secondary structures. I just said that, and I emphasize that again and again and again. Right, so here's a diagram of some hydrogen bonding between some two serines. Okay, so here's the serine, here's that protein backbone, here's the side group here, and here's another side group. So those two side groups can interact with each other via a hydrogen bond in the middle, okay, between that hydrogen and that oxygen. So anything with an alcohol or an amine on the end can form. And generally it's the alcohols, because the amines can turn into ammonium, alkyl ammonium ions, and so therefore have ionic interactions, but we'll deal with that later. Okay, so only certain amino acids will have hydrogen bondings with their side groups. That's all the alcohols and stuff like that. but only if they have those. All right, and I think that's it. Oh, sorry. There are many hydrogen bonds holding the protein in shape, whilst each individual hydrogen bond is not that strong. The combination of all of them combined, because remember it's thousands of these little bonds holding them together, can keep the protein in shape. And that's it. Adios.